Thank you. Let me just check that I make this on. Thank you very much. How's that for audio? Good. And if we could kill the front lights, is that possible? Thanks. Excellent. I'll now show you my first slide. Let's look. Yeah. Okay. So it's very pleasant to be here, and I still have no idea why I was invited, but it doesn't matter. I'm here as a sort of ambassador from another world, the world of old people, the world of statisticians, the world of biology. So there's a lot of translation going on here, and I hope that what I'm going to tell you is at least somewhat interesting. Obviously, I'm going to advertise. The speaker yesterday mentioned there's not enough statisticians and AI people and machine learning people to address all the problems that are around. Absolutely true. So we've got to seduce you away from the ones you are looking at, if you're looking at any, and get you into ours. So this is a bit of a pitch to say, come to an exciting field called biology, where models are very different from the way they are in physics and other areas, because they change daily. What's going on here? Am I doing something wrong? Oh, I don't want that. Can you kill that? Or is that necessary for the camera? No. Please, no. I don't want any lights. Thank you very much, anyway. Is it on? Yeah, cool. Okay. Being an old guy, everything you say is done by young people, which is not entirely true. That guy, Chris Woodruff, is almost as old as me, which is a shocking addition for him. Scott is an undergraduate, and Chris is a retired defence scientist, both working in my lab. So, I don't have a very big lab, and I'll say yes to anybody. So when a retired defence scientist says he'd like to join my lab, why not? And then he recruited Scott, who's just amazing, and I'll explain why later. Okay, so that's the two topics, and I hope I've said enough thanks to you guys for that. And of course, remember the quote that I mentioned at the end. When I worked in CSIRO in the 1980s, and I arrived, and everybody was doing modelling, and I thought, my God, what is this? And I created a discussion which lasted several months about what is modelling, because there are all sorts of weird people doing modelling in other parts of CSIRO. But then that quote that I gave, all thought and human communication is modelling. And the problem just arises when you mistake a model for reality, or you mistake one model for another. So do remember that as a take-home. All right, so I'm going to tell you a little more about me, but I've probably told you more than enough already. Then touch these topics very briefly. This is all incredibly superficial, but hopefully it will excite a little bit of interest. So, me. So this is a slower version of what I inflicted on you last night. That I like to begin with a question. Doesn't mean I always have, and I always will. This is already 7 years old, and I'm starting to wonder whether I believe it. I mean, I actually think you have to reinvent yourself about every year to keep your mind working. Real world phenomena, I'm definitely interested in the real world, though it is true, when I did my PhD on algebra, the real world was some distance. That's part of the reason I got out of algebra. And I like to use statistical models, but not invariably, as you will see. Well, I've got the word assumptions there, which I hate. What the hell? But measures of uncertainty. I brought this t-shirt out. I have an enormous collection of t-shirts, most of which I never wear. This is probably 30 years old, but at least it shows I'm on your side. Not that I heard the word uncertainty mentioned very often in this conference. I think you've kind of written it out of your history, right? Theorem is much more important, I can tell that. Most of you people want to be pure mathematicians. I know, I was one once. It's anyway, questions from the real world and data, head for answers and measures of uncertainty, and I see models as mediators between those two. But of course, now I'm in a world of science. You have to supplement this with various, what you might call diagnostics or graphics, things that tell you that maybe you didn't screw up. Plus, contextual checks. And I'm absolutely fanatical these days about controls. Don't ask me later if you want consistency with previous results, plausibility. So the last thing I want to do is say, trust me, I'm a statistician, here's your answer. That would be just ludicrous. You've got to link it up to everything else. Okay, end of PR. So this is the real world I live in now. Far left, genes or DNA molecules. Middle, messenger RNA, when you transcribe. I'm avoiding inflicting on you what is sometimes called the central dogma, because it's not very central these days. DNA, RNA, protein. And then proteins, which there are, and then metabolites. I also get involved in metabolites. For example, that is a string of DNA letters, which I'm sure you all know is a 4-letter alphabet, and it has a grammar infinitely more complex than the human, the sort of stuff that natural language processes deal with. Next up is the messenger RNA transcription from that, which is pretty much the same, except T's become U's. And finally, in that sequence, peptide, that's the translation of that messenger RNA, where you use the genetic code to translate blocks of 3. So we could have, and I'm going to talk later, about billions of these letters that we have to play with. I've got a prop here. I'll wave it down just in case I forget. There's a little machine that you can put in some cell material and plug that into your computer. And 48 hours later, you will have 10 gigabases of DNA. This is one of the most amazing things, which is the second of my topics. Okay, oh, by the way, metabolites, we were having a metabolite last night. If anyone recognises that chemical formula, it's one of my favourites. Alright, so the data these days, it doesn't come by somebody writing out a sequence of letters, it comes from machines. And up there on the top left is things called microarrays. I forgot to bring some, I usually bring a pile to show you, but you can imagine they're pretty small, they'll all sit in the palm of your hand. And along here, Illumina, HiSeq, these are DNA sequences, big machines, but that flow cell, again, it fits in your hand. And that'll be a gadget that will have billions of pieces of DNA in it, and you stick it in that machine and wait a while and you'll get out billions of letters. And down here is the MinION, the guy that I've just flashed, which I'm going to be talking about later. I'll give you a spoiler. Deep learning is used to analyse this data. And on the left there, bottom left, mass spectrometry. That's another way of generating masses of data about proteins. So, DNA top left, DNA messenger RNA here and here, and proteins there. And actually metabolites as well. Mass spectrometry, liquid chromatography, mass spec, or gas chromatography, mass spec. So, these guys generate huge amounts of data. And there's an enormous shortage of skilled analysts for playing with that data. Okay, on to my first topic. Single-cell RNA-seq. I've got to do a lot of translation here, because that, it would be silly if I didn't mention the word that people like me use, but I have to tell you what it means. So that's code for measuring gene expression in single cells. Obviously you understand the idea of single cell, but when you say RNA-seq, that means messenger RNA that has been sequenced, so you know what that messenger RNA molecule is, and you've got it out of a cell, so it's telling you what's going on inside that cell. At least which messenger RNAs. It doesn't tell you which proteins, it tells you which messenger RNAs. That's the intermediate between the DNA and the proteins. Okay, so everything these days has Moore's laws. I'm in a world where there's more and more data, and things get smaller and smaller, and so you've got a Moore's law working for you. See, back in 2009, that group, Tang et al., managed to get all the messenger RNA out of one cell and study it. Right? Now we're up here at 2017, 10x genomics. Well, I've never had 100,000 cells yet, but those circles, by the way, the red circles are technologies that I have had some association with. So I'm sort of going up Moore's law. There's another Moore's law for DNA sequencing, which is so old hat these days. But earlier in my life, sequencing was incredibly slow. One reasonable sized piece of sequence was one PhD thesis. And that would be like probably a millisecond of one of these machines now, that amount of data, but the way in which it was. So there's Moore's laws everywhere here. Things getting smaller, data amounts getting bigger. Alright, so I want to tell you now briefly about one project I'm involved in. So I'm interested in cancer immunotherapy. We're all interested in curing cancer. When I first went to my present job as I started the bioinformatics at this place I work at called UAI 20 years ago, and the director said, "What's your great aim in life?" And I said, "Well, I don't really have one. I just like to get a little more out of data than other people do." And she said, "No, it's not good enough. You've got to have a big aim." I said, "Okay, I want to cure cancer. How's that?" "Yeah, excellent." So now I've got a big aim. I want to cure cancer. Okay, now I'm sure you're aware of the idea of immune cells that sort of seek out the bad guys, like if it's been infected by a virus or bacteria, you want to kill them. Well, you want to do the same with tumours. And of course, if you get cancer, that means your immune system didn't work all that well. Didn't quite remove that tumour. Okay, so here's a serious question. And I didn't put it there just because I'm banging on questions. This is, you can extract the right sort of cells, because any tumour will have a cluster of immune cells around it. And you know, if you knew how they were thinking, you'd think, oh yeah, they're trying to kill the damn thing. They're not doing very well, because it's figured out ways of evading the mechanisms that they normally use. But maybe there are some cells in there that actually could do a good job. There's just not enough of them. And if we knew exactly what they were, and we could help them proliferate, tumour might go away. That's the sort of loose strategy of tumour immunotherapy. Okay? Now, with single cells, you've got a chance of doing this now. And it's really exciting. If you sort of suck off the cells from the tumour and look at what they are, analyse them, and say, yeah, these are the guys that are probably going to do a good job. Let's encourage them. So that's a project I'm involved in, and we're very close to putting together a first paper on this topic, and I'm going to show you some pictures. How would you know how to get some cells that might be promising? Well, you could take somebody with a really bad type of tumour who appears to be doing okay. And you think, well, if that person's got a tumour, that most of the time will wipe that person out very quickly, and they're still going. It might just be because they've got some good immune cells helping them hang in there. Okay, so you take a near-death patient, or a patient who would have a very bad prognosis, suck off the immune cells and do what I'm about to show you. Okay, so top there, sample summaries of 5,000 cells from one tumour. This is patient 1 in the paper we're writing, all referring to messenger RNA molecules, which we loosely call genes, just for, we call them genes and you think of them as genes. Top left, total molecules counted per cell, median 1700. So this is a very low coverage, right? I mean, a typical cell would, well, we don't quite know exactly how many of these molecules would be in a typical cell, but it's probably 50 to 100 times that number. But it comes down. Down there, number of genes seen per cell. We look at all these molecules and we say, what gene are you from? Because there's 20,000 possible genes for protein coding messenger RNA. Okay, there's 20,000. We might see on average about 1,000 or a bit less. Which means if you divide one number by the other, we get about 2 molecules per gene. Of course, we get a lot of some, and we get very few down to 1 of others, and of course we get millions of zeros. Look at the top there. Expression of gene 1, expression of gene 2, gene 3, gene 4. Look at the first bar, the zeros. 4,000 out of the 5,000 was 0. And then there's this tiny little hump there. And that tiny little hump is not, big numbers. You see, we've taken the number we got and multiplied by 10,000 and divided by the total. That's a kind of normalising in our jargon. But at least you can see that in this panel on my right, the bottom left there, that's a reasonably abundant one because it's only missing 600 times out of 5,000. Whereas the others are like 4,000, 4,000, almost 4,000. Most of the time you get nothing on those genes, but you get plenty there. So, 20,000 genes, each of them has a picture like that. I can't show you all of them, but I want to rub your nose in a bit of data because data is our bread and butter. Okay, so that's the start. Okay, some aspects of single cell RNA-seq. High dropout ratio. That is a chance rather than real zero. And that's one of the interesting things about these guys. Some of the genes will not be expressed in that cell. But when you sort of pull out what you can get and look at it, you'll get a lot of zeros, which are just by chance, because you took a small sample. On the other hand, there will be some real zeros that are not actually there, no matter how much you took. We have to worry about that. So there's issues of normalization. Big cells have more molecules. Small cells have fewer. We get less data out of some. There's a lot of technical variation you have to deal with, and there's a lot of heterogeneity. You know, if I'm taking stuff out of a tumour and looking at it, you'll see in a minute. You've got a lot of different sorts of cells. Okay, I'm going to now very quickly mention some of the analytical challenges. Top left, we're first of all cell level analysis. I've got 5,000 cells, so we think of the cell as the unit. Well, obviously, you know, people cluster. You cannot stop people clustering, no matter how much you try. So I gave up a few years ago. When I was a student, clustering was very long to notice. Like statistics was good stuff, right? Models and you know, parameters. Clustering, give me a break. That's the sort of thing computer science did. These days, it's indispensable in this world. Now, visualizing your clusters is pretty good, pretty important. So I'll mention that. So there's many, but you need new tools. If you're going to have 5,000 cells and a small, let's say on average 2,000 of 20,000 possible genes, this is not necessarily going to be a thing you're going to use k-means or some standard hierarchical clustering. You have to think a little carefully. In other words, even clustering has to adapt to the type of data. Looking at it, and this is a cool problem on the left here, cellular trajectory inference and branching. I've got 5,000 cells. They may have structure within them, which is not clustering. It could be a temporal pattern, because they might be different ages. They might be at different stages in some developmental cycle. Can we distill that? Well, that's a really interesting problem, and there's probably more than 10 new tools now. I don't think this problem was being addressed, at least not in our world, 5 years ago. So here's a great example from some of my friends in Berkeley that published a couple of months ago. So we're interested in the mouse nose, also known as the olfactory epithelium, where all these molecules floating around in the air, they come in and they stimulate these olfactory receptors, and it's thought that roughly one smell, as it were, one molecule is associated with one reconfigured olfactory receptor. So there's the mouse in his nose. So of course, you're going to execute the mouse, pull out all the stuff out of his nose, and have a look at it. And on the bottom left there, that is a TSNE, you know, T stochastic neighbourhood embedding picture of all the clusters that were found in that sample of cells. You know, we've got some green ones and some cyan ones and some yellow ones and brown ones and orange ones. And their challenge was to say, do we have the information of a developmental pattern inside those clusters? In other words, we're going to try to draw lines through the clusters. So this thing on the top right is what they inferred. We start off with stem cells, obviously. Pretty much any of these sort of things begin with stem cells, whether it's blood or cells from the olfactory epithelium or cells in the brain. You've got neural stem cells, you've got olfactory stem cells, you've got hematopoietic stem cells. Everything begins with stem cells, which, of course, self-renew. And then we've got a bit of signaling, we've got some brown and then grey. So you can see up here, green, the stem cells, brown guys, grey ones, so there's a kind of a line there. And then we've got a bit of a dead end there up to sustentacular cells. These guys here. Whoops, this always happens. These guys, whoops, oh yeah. Anyway, I'll just point, right? The red things, sustentacular, they are support cells that help this epithelium stay together. So they're off there, and now we've got this grey line coming right down to the olfactory sensory neurons. They're the guys that do the smelling for you. And we've got another side branch there to the microvillis, which again are structural cells. You see the little diagram up there, the ones that sort of hold the structure together. So this developmental hierarchy was inferred using an algorithm called Slingshot, which was developed by a student of Sandrine Dubois, who edited that book we heard about. Well, they think it's right. I mean, they do all sorts of biological checks to convince themselves this is not just an algorithmic fantasy. This is a legitimate developmental, we've got some developmental lines that go to the structural support cells, others that go right down to the olfactory epithelial cells, these guys here, that do the action. Okay, so that's a really interesting, I think, analytical problem. Now we'll go to gene level stuff. Well, the really basic gene level thing is you just compare two cell types that you knew already. That's not very exciting, but you need a whole lot of new tools, and I'm going to mention one. But then there's a few sort of crossover things. You see clusters on the left, you're going to need to find local genes that distinguish the clusters. So you've got to replace cell type by cluster up there. If you've got a developmental pathway that puts a kind of time, a multidimensional time, or a tree-like time on your cells, you now want to watch how genes track along those times. So we've got a gene level reflection of that. Gene-gene correlations is really challenging. If most of your data on individual genes is 0, you can imagine that most of your data on pairs of genes is 0, 0, or 0 something, or something 0, which is kind of hard to use to estimate a correlation coefficient. So, and there'll be a little bit of something, something. So that's a hard problem, but people are working on it. And imputation. Obviously, a lot of these zeros are only 0 because you missed out. There was data there, but you didn't get it because of your sampling. Back to our tumour cell, our tumour samples. So if we look at the left first, this was clustering done by a package called Surat. Anybody who has a deep artistical education will have heard of George Surat and know the connection between Surat and dots. So that's a clustering method which is being visualised by TSNE. Right, so TSNE is not the clustering, TSNE is the visualisation in 2 dimensions. And as you can see, we've got 10 clusters there, all coloured from 1 to 10. And the first thing you want to do is say, what are these clusters? Because we expect most of them, we know. There'll be CD8 positive T cells. There'll be maybe, just name some sort of subset of the white blood cells. CD4 positives. Of course, we have to select these cells, so we know roughly what we expect. But what we're hoping is there's a cluster that we didn't know before that we can identify. So how do you identify them? You use what we call marker genes. A marker gene in our jargon is a gene that you know something about. Like if you've got a cluster that you think might be CD4 positive T cells, well, you've got to know some genes that are up in CD4 positive T cells and down in others. That'll be a marker for CD4 positive. And traditionally these are on the surface of these cells and they're used to find these cells. But we're going to find out stuff inside as well. But remember, the goal is to find a cluster of cells that you didn't know about before that look promising for killing that cancer. So here is 6 markers, and you can see if I go up there, this marker is highly abundant in those cells there, so it tells us something about that cluster. This marker here and these 2 are highly abundant in that cloud. So it's telling us something about that. So you sort of roll out 10, 20, 50 genes, have a look at where they are, and say, yeah, well, I think this is that, this is that, this is that, this is that. This one looks very interesting. And that's how we did it. Okay, so the marker genes are where we know which cell types they're high and low. Now, you've done all that, you've figured out as much as you can about the clusters, you've got this fantastic new one. What's in it? So now we're going to do differential expression between that cluster and the other guys that we think we know about. So that's where differential expression. What genes are up in that cluster or down in that cluster compared to others. And I've got one slide here which I'm not going to take you through, but this is a standard parametric statistical model problem. And I just want to tell you, I do not think that God generated the data by running that model with some noise and presenting us with the numbers. This is a model that is to help us answer the question, what genes are up in one cluster relative to another, and what genes are down in one cluster relative to another. So it's what we call a zero inflated negative binomial. We've got more zeros than you would expect, by chance. People that know about negative binomials would know that they are unimodal. So we better not do this where we have 10 clusters, because the gene might be up in some and down in others, and it would not be the slightest bit unimodal. So this is where you're down to 2 clusters where you think are reasonably homogeneous. And we have a dummy variable there, x, to tell us whether we're in cluster 1 or cluster 2, and that will mediate the mean parameter and the zero inflation parameter. And then we crank the old EM algorithm and away we go and get some estimates. And we have our favourite data sets to try to test this on, where we know the answer, and we end up finding the genes that are in our new interesting cluster compared to everybody else. So that's how it's working, and it has worked very well for us. Alright, so that's as much as I'm going to say about single-cell RNA-seq. There's a lot of other single-cell stuff going on. I mean, obviously there's DNA in single cells. Let's look at that. I was talking then about messenger RNA. There's proteins in single cells. Let's look at that. Well, that's what this is. I'm not going to say any more than flash this slide up, but the buzzword is cytoph. Cyt for cytometry for cells, top, time-of-flight mass spectrometry. A wonderful technique invented and perfected by a guy called Gary Nolan in Stanford University, where you can look at 40 proteins in millions of cells. So if you like multivariate analysis, have you ever had 40 dimensions where you've got millions of data points? I mean, this is just amazing. And a lot of very interesting things. Some of the things I've mentioned already, TSE plots and clustering and temporal evolution, developmental pathways. There's so much you can get out of this data. Because remember, and these are all good measurements, the 40 good measurements. I was talking before about 20,000 measurements, most of which were zero on each cell. Here we've got 40 measurements on each cell which are good. And there's millions of cells. So this is sort of a new world, and anybody that loves lots of data should go berserk with this world. All right. That is, I have to check the time. 9:04. I start about at least 8:35, maybe even 8:40. Yeah, okay. So I'm sort of, half-ish way through. Are there any questions, by the way? That's a good way to slow down. Any questions? What if I told you I'm not going to continue until I get a question?
You have lots of data, but presumably you also have other information, like information from other experts and biologists and other people. How do you incorporate the expertise with the data?
Okay, well, at the risk of self-publicity, I'll go back to slide maybe 4. Yes? Look at those bottom lines. We do our fancy so-called analysis, and then we have other checks, controls, consistency, plausibility. So that involves things like talking to the biologist. What do you think of our results? Does this look right? Does this look wrong? And they'll look and say, well, this shouldn't be up. There's something wrong here. Or, have you got a control? This guy should not be changing. Or this guy should be there and it's not. Why is it not there? And so on. Does that fit the description you're talking about? Yeah, so this is the, you could loosely say this is the important part of the analysis. After we've done our magic and given them our measures of uncertainty, our p-values or our confidence intervals or our blah, 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 then the question, does any of this make sense? And that is arguably the most important part of the analysis, which involves a lot of interaction with the biologists.
They analyse the output of your data, they don't.
They look at it and they say, where is X? And we have to say, well, actually, X, we didn't get very many counts from X. Mostly it was 0. What's going wrong? Better do some imputation. See if we can find out whether X should have been there and we just missed it. So that type of thing is a constant interaction once we produce some interim results. And it can be quite tough because our analyses don't always deliver what they would like. And sometimes they're right and sometimes they're wrong. So, that's interaction. Thanks for the question. Any further questions? Yeah?
Harry, I come from the instrumentation of measurement. The problem, you have questions about data. The data is really tricky because you have to get the instrumentation to measure the data.
Oh, yeah.
And then you come to the uncertainty. How about you go to the reverse way? If you give a certain uncertainty to get the correct answer, then particular questions, that how do you define the intolerance for the data approximation, that not to give you the guidance, how do you design the instrumentation?
Okay, that's a long and hard question, but it will prompt a couple of answers, of course. One is this. That little dream world description I got there, idealized means dream world, right?
Yes.
Not reality. Often the data come first. Like people read, oh, we've got single cell RNA-seq. Then they think, what questions can we answer with this data that we couldn't answer before? So it's like data drives questions, then they collect the data, then they come to us. Only when it gets more mature do we say, okay, now we've got this data tight, we've got this capacity to generate this data, and we would like to address this question, can you help us design a study? But that's at what you might call the semi-mature stage. In the early stages, wow, look at this data, cytop data, single cell RNA-seq data, this sort of data, that sort of data. Go crazy thinking of all the things you can now do with it. This is the biologist, that they couldn't do before. But somebody had to figure out the technology, the instrumentation to measure that. That's usually not us.
Okay.
Right? We are users, we're not developers, at least in this field. Other people, I mean, we have an image analysis group that are very active in microscopy, trying to get to the frontier there. But this sort of stuff, this is, if you like, important technology, and we get roped in because the biologist sees the capacity to address questions they couldn't address before, like the immunotherapy, right? If you want to know, what do I need to treat these cancers? What immune cells are going to knock these cancers out? This gives you the capacity to do that. That didn't exist 2 years ago. Okay. Got a question or push on?
So if we go back to the picture of the clustering and then the process that the people at Berkeley got out of that, I'm looking at this one. I'm wondering, how do you fight the natural phenomenon where people had some notion of what the process should be? Like,
Yes.
And then you try all kinds of clusters, and one of them will be closer to the process that you wanted to get. Right? But overfitting in the exploration of models. So how do you fight that as a field?
Well, I think, I'm not saying there is any simple answer to that, but these sort of techniques would be firstly dreamed up and then tested on simulated data where you built in the developmental stuff and found that you could more or less get the right answer. Then you will apply it to some quote-unquote real data of interest. And then you will, like I said earlier, scrutinize the result. So I don't think it's a device for steering towards what you want, right? You're not going to get a paper. That paper in bioarchive has been submitted to some journal, probably Nature Methods. They're not going to accept it unless you've got some very convincing evidence, typically 3, maybe more, independent data sets where it has delivered what you know. For example, you could apply it to blood cells where pretty much people know most of the major developmental pathways in blood cells from hematopoietic stem cells and some other ones, right? So you give it some credentials, then you apply it to a new context. And I mean, in a sense, that's how we do any of our statistical analysis, right? How do I know what I did with this data is right? Well, I don't. Even if I've got some beautiful measures of uncertainty. But the thing is, if I've found that it delivers things that are known to be true in a reasonably objective analysis of several data sets, then that is, it's what you would call transfer learning, or something like that. You've got some buzzword for this, right? No, anyway, you've got some that if it works in 1, 2, 3, 4, 5 contexts, maybe it worked in the 6th. And of course, you'll get a check that you didn't screw up because it's not good to have a paper in stem cell which gets the wrong answer, and then your competitors are going to deliver this in their rebuttal. Okay, so that's sort of how it works, I think. All right, more or push on? Yeah, so we've solved the cancer problem, and we're moving on to. And actually, we're doing a lot of this stuff with the treatment of drugs, because you can only measure 40 proteins, you have to choose them very carefully. So we're interested in particular parts of cell machinery, cell signaling, and so you have an appropriate set of proteins, you need to develop antibodies, they've got to be labelled with these amazing elements. I can't tell you how interesting this technology is. All right, at this point, I want to switch to this gadget here. This is a Nanopore, long read sequencing. Here's the translation. Long in this world means from thousands to hundreds of thousands of these letters. One long molecule, let's say 100,000 base pairs long. And you've got the sequence of letters for all of that. That would be called long in our world. Current technology that I mentioned earlier in the top right, the Illumina, might get 200 at a time. So you get lots and lots of short sequences. This gives you lots and lots of long sequences. A read in our jargon is a DNA fragment. You just call it a read because it's stuff that goes in and you kind of read it. Sequencing means you estimate the bases, or you infer the bases. Okay, so that's what we are talking about here. I failed to get my video embedded, so I'm going to have to go out of PowerPoint, show you a video, and go back in. Hopefully you will be patient. So, let's make that big, and let's run it. This guy here is called a nanopore. Well, of course, it's an artist's impression of a nanopore. And those things going down it, that's meant to be electrical current. So I suppose we're supposed to think of those guys as electrons. And occasionally a big molecule tries to go through. And of course, we're measuring the current as it goes, right? So we've got stuff pouring through this pore, excuse the pun, and there we go. And now we're going to start sticking DNA molecules through the pore. And there it is. And of course, they get ratcheted through. They're sort of attracted through by a voltage difference. And as they go through, depending on whether it's an A, a C, a G, or a T, you're going to have a different kind of obstruction. You want to see that again, or is that enough? I could show it once more. That's probably the best part of the talk. So let me repeat it. Nanopore, little pore. Whoops. And by the way, that thing, this is it. And you plug it into your PC or Macintosh idea, and all that data collects. And it's a serious amount of data. I'll just tell you a little story. This guy, Scott, the undergraduate, who was the co-author of a lot of this stuff, they had things they call pore camps. You know how you have little camps now where everybody sits around and looks at a program and analyse things? We have those in Australia anyway. And a pore camp is where you sit around and talk about nanopores. Anyway, everybody at this pore camp got one of these guys, and they stuck some cells in there and they run it. And to a person, everybody's computer crashed. And Scott went home that night and wrote a data compression algorithm so that when I came back next day, the pore camp could continue. This got reported on Twitter, and he got an email from a journal saying, hey, we'd like to publish your method of compressing, and he wrote it out, and it got published about a week later. Some incredibly short time from pore camp, idea overnight, Twitter, journal, accepted. It's not a world that I live in, but I can observe it. Okay, so that's the nanopore data. Back to PowerPoint. So, yeah, that was supposed to come on this slide, but for reasons known to understand, it didn't work. Oh, but anyway, 450 bases per second. That is, you're putting this DNA molecule through, and I don't know whether I said it back there. I did, but I'll probably say it on the next slide. Yeah, so it's 16-bit measurements, and it's running at 4 megahertz. Right, so we're getting 4,000 measurements per second. So there's 450 bases a second. So you're getting on average about 9 measurements per base. And there's a sequence of raw data from this guy, putting some E. coli DNA through it. In the ONT, chemistry is evolving rapidly. ONT means Oxford Nanopore Technology. And so that's the data. That's a 60 millisecond time sequence. There's about 240 measurements there, and there might be of the order of 30 to 40 bases in that data. Okay, so you've got to segment it. That's not like a serious analytical challenge. I should say that the idea is that a cluster of numbers corresponds to, you know, it's sort of stuck there for a while with one particular letter, one particular base in the operative region, and then it moves on to another one. So the level jumps up and down. One question is, what's driving that? Is it the actual letter? No, you can see more than 4 levels there. Is it pairs? No, you can see more than 16 levels. Is it triples? No. It looks like somewhere between 5 and 6 kmers of 5 to 6 in length is what's driving that height, that current strength. But there's also some long-range stuff. So, would you believe it? We have the opportunity to use long, short, blah, blah, blah. All right. Anyway, you segment it, and then you start trying to call the bases. That is, you start to try to read off the letters. And I'll just very quickly mention 4 algorithms that are there. That's pretty much all that's there. You'll notice almost all these dates in 2017, which shows that this is like super recent stuff. Minno and Alvapor are run by the company. We don't quite know what's going on. They started off with 6 HMMs. So, you know, you have a level for every one of these 6, you move it along, and so on. It didn't do very well. They now use some recurrent neural network. Details not available. Nanopore still uses sigmas as a state, and I'm sure you can guess how, you know, you've got these sigmas going through. For every sigma, you're going to have an emission distribution, which will be an average level or a standard deviation. And after you segment, you're basically trying to work out which sigma is in the system as a result of the level. Deep Nano, would you believe? There has to be one, uses deep learning. They use a bidirectional RNN, and base crawler is even more complicated and probably even better in some ways. This is close to a real-time crawler. In most of the others, you need to have all the data, and then you call it. This guy calls it with a very small time lag, and they use a 2-step process where they use stacked, long, short range, I've forgotten what the T is there. Anyway, you know what I'm getting at here. Okay. And yeah, I won't give you the details, but suffice to say, this is my introduction to your world of deep learning, because these algorithms work. They deliver letters more accurately than things I understand, like HMMs. So, as we'll see in a minute, I want you to keep working on helping me understand these damn things. Alright, so this is like some final output. We have, this is now not in the time index, but event index, so those little gaps are equally spaced, and we get a string of letters out. So this guy has 512 pores, so each pore is running, you know, as I mentioned, 4 megahertz single, and roughly speaking, 450 bases a second, and the idea is you put your sample in there, and you plug this in, and you let it go for 48 hours. And all going well, most of these pores will work. Sometimes they get blocked, and you might get 10 gigabases of data from running this single thing in 48 hours. And it's meant to be so you can go into the wilds of everywhere, you know, like go to the backlocks of New Guinea and take a blood sample from a person who might have malaria and stick the blood sample into this machine and wait for 48 hours and have a look at what sort of stuff's going on. Or viruses, or you can imagine the portability of this and the depth of information. In case you don't understand, did I say 50, 10, 20 gigabases of data? This is a serious amount of data. Our entire human genome is 3 gigabases. Okay, so this is an artist's impression just to show how overwhelmed I am with all this. So I just think of this as a black box. I have no idea what the hell's going on, and I'm hoping, and I was pleased to learn a few other people here have no idea what's going on as well, which is very reassuring, because I sort of think, oh, here we're dummies sitting in the medical institute, and all these other people understand RNNs and LSTs and bidirectional. Anyway, so there's a lot of stuff, and of course, I wouldn't even know how many parameters. I'm not on top of it. Okay, so I'm now going to go to the last aspect of my talk, 9:21, so I'll probably, yeah, this is 10 minutes and I'll shut up. But is there a question about that? Because I'm going to talk about something slightly different which uses that technology in a minute. But I'll take a question about the technology or about the data or about the algorithms.
Just curious, but I mean, what if there's so many same letters. So really, segmentation, I mean, that seems to be also something where there could be challenges.
Absolutely. I mean, for any type of sequencing whatsoever, when you have what's called a homopolymer, that is the same guy repeated several times, we had some in that picture, didn't we? So there's a string of A's. Now you might ask, don't we make errors when it's a string of A's? And the answer is, of course. Most of the errors are like that. And that's actually true for most sequencing technologies. That distinguishing 7 A's from 8 A's or 10 A's from 12 A's is hard. Okay? And that's been so for approximately 40 years. Almost since sequencing technology began. And there's different reasons for different technologies, but basically you're measuring the length of the molecule in some way, or the duration of time that's kind of stuck in that little pore before it comes through, and all of these things get tricky. Obviously, if you're looking at sigmas, and the previous sigma is the same as the next sigma, how do you know it's not, how do you know it just didn't stay there a little bit too long one time, a little bit too short another time, and so on. Thanks for the question. More questions? Up the back?
What's the benefit of getting the exact sequence? And can you correct for error based on some, like, if you have some baseline or some biological knowledge of what the sequence?
Okay. Everybody here, the question was, what's the benefit of getting the right answer? Good paraphrase? Okay, well, there's two. One is you may not know what it was to begin with. So intuitively, if we're doing human stuff, then whatever you get could be full of errors, but it is very long, and you map it using fancy algorithms that we have in bioinformatics back to the human genome, then you should be able to sort out the errors. And that's true, obviously, for E. coli and other things. So this is used when you want to train it. But obviously, you do want to do it where you don't know the answer. And one of the reasons this is so fantastic, we don't really know what the human genome is. All we know, it's a bit like what I was saying last night, that this guy says we developed the Mars Explorer to land not on Mars, but on a model of Mars, right? So what we call a human genome right now is just what we've got from the technology we had to now. And things like repeats, and actually, 50% of the human genome is repetitive. Could be single base, could be pairs, could be triples, could be 1,500 base pairs repeated around the place. So we do not have a decent human genome which covers the repetitive regions. This technology promises to get you there because it's so long that you might be able to span the entire repetitive region. Whereas if you're doing 200 base pairs, you map that to the genome, it could go to 57 places, so you just throw it away. So the current human genome is not even complete. This is going to complete it. Then we'll discover how much your genome differs from a reference. And then there's all these other weird organisms, bacteria and viruses we have no idea about. We want to get the right answer. Is that good enough? But for many purposes, you're absolutely right. You're dealing with a known organism. You just want to get quick data, map it there, sort out the errors, and find out what's going on. And for my next part of the story, that's quite adequate. Other questions? All right. So, methylation. I'm going to inflict a little bit of chemistry on you, not too much. But there's a chemical modification of these ACGTs called methylation, which, would you believe, adds a methyl group to a certain place. And what is called 5-methylcytosine, cytosine is the C in the ACGT, and then at position 5, starting 1, 2, 3, 4, 5, you can add a methyl group, CH2, and it will be modified. This is very important. And so we just think of it as the white C going to a yellow C. Or you can think of it over here as it lighting up like a light bulb. That's the double molecule you see. So if you've got a C followed by a G on one strand, you'll have a G followed by a C on the other. And most of these Cs are methylated when they precede the G. Okay, so that's an important thing. The next slide is supposed to convince you of that. There's at least 6 things that are relevant. Chromosome imprinting, I'll explain. X chromosome inactivation. Women have 2 X's, men have 1. We do not wish them to be twice as strong as men. So nature has evolved the way to cut the women back to the men by silencing one of the 2 X's. So that stuff that comes off the X the women do not get twice as much. This would be a serious challenge for male supremacy, so unfortunately nature solved that many million years ago. There's other reasons. Transposing elements. In all this repetitive stuff, there's bad guys that jump around. We want to keep them dead. We don't want them jumping and landing in things and giving you ill health. So, repressing them. Aging. I'm very familiar with it. You will become familiar with it. Methylation is an important component of aging. Cancer. Genes that should be on and are not. Genes that are not on that should be. Methylation plays a role in that. That's 5 things, and much more is 6. I want to tell you what imprinting means. There's a sort of a canonical story about, it goes back to Mendel. There are little things called genes. Mum produces some and dad produces some, and they get together into the child, and then so on. So they sort of pass down the family tree. You don't normally think that when I've got a bit of a chromosome from my dad, that it has dad marked all over it. You just think it's a molecule, right? And similarly for mum. So they're just mixed up together. We have 2 copies of all of our non-sex chromosomes. The guys have 1 X, and which they definitely got from their mum, and 1 Y, which they definitely got from their dad. But the others, you don't necessarily know where you got them. But imprinting is like a stamp that says, this little bit here tells you, yeah, this is actually a maternally inherited chromosome. And this bit is a paternally inherited chromosome. And it's methylation that does this. And it's essential for life, in the following sense. That if I take DNA, I could take a full complement of chromosomes, all from a guy, and put them in. See, that's androgynote. That's like the entire set of chromosomes for that individual there comes from men. Doesn't work. Will not develop. A gynogynote, same. Women only, sorry folks, it's not going to develop. We need chromosomes, 1 from mum and 1 from dad. And the markers are these imprinted genes. Just bits of the DNA that are sort of stamped paternal, maternal. And it's a very interesting and important phenomenon. So we're going to figure it out using the long-read stuff. How do we do it? Well, M means methylation. So if you look there, there's a C preceding a G. The signal for that guy, I write it as a triple, but in practice, think 5 or 6-month. It's a little bit different if that C is methylated. Because it's sort of like you're dragging it down through the pore, and the methylation is a little ball or whistle hanging off it. It's just going to slow it down or speed it up a little bit. So you'll get a different signal whenever the C is methylated. And that is enough to figure out not just whether it's A, C, G, or G, but whether the C is methylated. And that's really, really cool. And getting close to the end, I want to mention a few buzzwords that will make you feel warm and fuzzy. HMMs you've already heard of. How about variable order HMMs plus hierarchical Dirichlet process mixtures? We heard about those with lions, didn't we? Was that right? We had some lion data and we're inflicting a hierarchical Dirichlet. Actually, there was a hierarchical Dirichlet mixture of HMMs. So it's kind of a synthesis of the top 2. We've got a picture there which I won't explain. And then there's now raw. So currently there are 3 algorithms out there that will call methylation. And they do make use, whoever asked that question, of mapping back to the genome first. And then we do a post-processing to see that where we had a C is actually slightly different. And maybe it's a methylated C. And there are other modifications I'm not talking about. Okay, so it's great. And we made use of Jared Simpson's nanopolish in the picture I'm going to give you, with 2 more slides and we're done. So, first off, take a lot of these long reads. This is a schematic, because those reads are typically thousands, and they might have tens to hundreds of C's that could be methylated. So the N there means it's methylated, and the C means it's not. Now, in reality, we don't have enough accuracy to call it deterministically. We just have a log likelihood ratio of C methylated versus C non-methylated. But it's pretty good, just not perfect. So that's some data where you call the methylation state. And now we're going to look at it in 2 ways. One is we're going to look across those All of those guys are aligned and mapped to the same part of the genome. Unfortunately, you can't see the letters there, not very well, but you can see here, for example, here, 50% of those are Cs, 60% of those are Cs, 2/3 of those are Cs. So, often people have been looking at methylation cross-sectionally for a long time. What this technology enables us to do is look longitudinally and see what's the methylation right along the very long molecule. And now you see we've got averages. We've got these guys here. Whoops, sorry. We'll go look at that. Oh, okay. Okay, so we're looking at longitudinal methylation now, and our interest is in whether some of these very long molecules are pretty much methylated in their entirety, right across. And there's one extra detail here. Every chromosome is in our, apart from the sex chromosomes, has 2 copies, one from mum and one from dad. And we can actually sort out which they are in this data, because it's 2 strains of mice that were crossed, and we can tell from the DNA, not the methylation, which strain it came from. So these 9 strands have been reorganised into naturally and paternally inherited strands. And my last picture is this, which is just mind-blowing for people who do epigenetics. We're writing this up right now. You can see that, you look at the top there, CPG Island is an area where there's lots of Cs followed by Gs. And they're the guys that typically get methylated or don't get methylated. It's a higher density of Cs followed by Gs. And the pink is the maternal, as always, and the cyan is the paternal. So what you can see, and this is a sort of average methylation across the entire roughly 20,000 base pair molecule. And you can see that this is rather clearly differently methylated. And this is a gene area that is imprinted, which means it's got the stamp of mum or the stamp of dad. And we can see this at the whole molecule level, at a 20,000 base pair level. Never seen a picture like that. You will not have seen a picture like that. We're hoping to get it published before anybody else. Alright, so that's where we're finished. I hope I've given you a bit of an idea that there's some fantastic biology here, but there's also amazing challenges for people that like a lot of data with interesting questions. And it is also true that the 2 parts of my talk have actually been joined together very recently. People can do long-read sequencing of single molecules. That's it. So this is my institute. The green we can see, by the way, is the oval of the school I went to. And it's kind of embarrassing to admit that my office is there. And I look out over the school oval that I went to 60 years ago. But, okay, single cell RNA-seq, Argus and Cheng Zhong are the 2 people working with me, and Peter and Sharina are the cancer biologists, and Christine, Matt and Lee are other people who helped. The nanopore sequencing, this amazing undergraduate, Scott, who's just with us 1 year, but he sort of dragged us into the world of deep learning. And Chris, the retired gentleman, you probably know Chris there, because he also works with defence. And other people who are involved. Nani is an epigeneticist who's absolutely top-rate methylation and epigenetics person, and other people. Thank you, folks.